this technology can contribute a lot to revamping our industries, contribute to food security.
It was identified that this maruca, pod-boring insect, is causing a lot of damage to cowpea. And this one is more pronounced of prevalence in Africa.
If the insect feeds on this new cowpea, which is improved genetically, the insect dies immediately, and so it will not survive, talk less of shedding other generations of the insect.
The normal one we have been growing, we've been having a lot of problems along the line, which especially during spraying. We spray up to 7 to 8
The
times.
Bacillus thuringiensis gene, also known as the
farmers
BT
have
gene,
been
is a
interested.
naturally occurring
They've shown
bacteria
their interest,
that produces a
and they always have asked us, where are the seeds? Where are the seeds? When can we have these seeds? The farmers cannot wait to plant these seeds. They are
Nigeria has what's called freedom to innovate, freedom to advance, freedom to produce new products. So Nigeria is, as a matter of fact, taking the right step, and Nigeria is going to move ahead.
It was identified that this maruca, pod-boring insect, is causing a lot of damage to cowpea. And this one is more pronounced of prevalence in Africa.
The normal one we have been growing, we've been having a lot of problems along the line, which especially during spraying. We spray up to 7 to 8 times within the season.
If the insect feeds on this new cowpea, which is improved genetically, the insect dies immediately, and so it will not survive, talk less of shedding other generations of the insect.
the farmers have been interested. They've shown their interest, and they always have asked us, where are the seeds? Where are the seeds? When can we have these seeds? The farmers cannot wait to plant.
Nigeria has what's called freedom to innovate, freedom to advance, freedom to produce new products. So Nigeria is, as a matter of fact, taking the right step, and Nigeria is going to move ahead.
insect. The gene inside the bacterium responsible for producing the beta gene toxin can be transferred. The Bacillus thuringiensis gene, also known as the beta gene, is a naturally occurring bacterium that produces a protein toxic to certain types of insects. The gene inside the bacterium responsible for producing the beta gene toxin can be transferred to crops, thereby making them resistant to corresponding insects.
Beta gene is a gene that enables an organism to protect itself against some group of insects. They are called lepidopteran insects. These are chewing insects, and is restricted to only those ones. So any plant that has this gene in it will be able to protect itself against this invasion of these insects.
In Nigeria, the beta gene has been used as a solution to problems of cowpea farming and that of cotton as well. For cowpea, the beta gene has been utilized for the crop's resistance to the maruca insect, also known as a pot borer, which preys upon the cowpea plant. And in cotton, the beta gene gives resistance to the crop against the pink bollworm.
You know, in every society, modification is the best. And when you modify your crops in terms of agriculture, it means that you are aiming for certain qualities and certain benefit. Part of it, when you modify it, is getting high yield, circumventing diseases that are affecting the crops, circumventing the insects that are devastating the crops. So all these have been the focus when we modify the cowpea and when we modify the cotton.
The integration of the beta gene into cowpea and cotton production in Nigeria stems from the gross damage incurred by farmers of the two crops for years. In the case of cowpea, the destruction caused by the maruca's infestation of cowpea plant results not just in reduction of yields, but also the quality of harvest, as losses could be up to 80% in severe cases. For cotton, on the other hand, the infestation of the pink bollworm on cotton plantations has made farmers lose tons of harvest, making farming the commodity a nightmare for many. The use of conventional insecticides to control these pests are not only expensive, but the availability is limited. Farmers also lack proper skills in their usage, thereby posing safety challenges. This narrative began to change in 2018 with the National Committee on Naming, Registration and Release of Crop Materials, also known as the Varietal Release Committee's approval for the commercial release of two BT cotton varieties. The approval for the commercial release of the BT cowpea variety followed shortly in 2019.
Reduction in insecticide spray and then high yield. It gives you high yield because they are already protected from the insects, those insects. In the case
Good morning, everyone. My name is Francis Kusodi, and I'll be emceeing this event for the Ecological Society of Nigeria. Let us start this conference by an opening prayer. Can anybody give us a match? Please give us an opening prayer. Okay. Okay, yes, sir.
In Jesus' name.
Amen.
Father, we are grateful this morning for this opportunity and for the privilege. We ask that you take absolute control of this meeting this morning in the mighty name of Jesus. We commission this meeting into your hands in the name of God the Father, in the name of God the Son, in the name of God in Jesus.
Let us also take the national anthem. I don't know whether we take the new or the old national anthem. I don't know. You can take national prayer. Okay, please. Please start. Thank you. Good morning again, once more. I'd like to welcome you to this conference, pre-conference. I would also like to open or call the chairman of the ESN, Professor Waiki Malak. All of our dignitaries are seated here, and everybody would like to welcome you to this program. So we'll go straight to the opening remarks. This will be done by the chairman of the Ecological Society of Nigeria. Please kindly welcome the chairman, ESN.
Good morning, everyone. The DG and Chief Executive Officer of the National Biotechnology Research and Development Agency, who is our host, many of your staff that are here, members of the Ecological Society of Nigeria that are here, other well-wishers, the presenter of the lecture today. I'm delighted to stand before you this morning to welcome you to this seminar. As our tradition, the Ecological Society of Nigeria was formed sometimes back in 1965. And right from that time, scientists have been interacting with insects and their allies in different forms. And I believe all of us gathered here this morning are scientists, and we have the quest within us to know what is happening to insects, the world of insects, the environment, insects, public health, agriculture, and so on and so forth. This year, by the grace of God, the annual general meeting, annual conference, which is the 53rd annual conference, and the HBN of the Ecological Society of Nigeria is going to be hosted by our branch. That means the Ecological Society of Nigeria, Abuja branch. We are hosting this event between 3rd and 7th of October 2024 in the University of Abuja. So as a prelude to the conference itself, we deem it fit that we start this way by gathering scientists around us, a cluster here, to gain some insight as a pre-conference in preparation and mobilization of ourselves and others that are not entomologists or entomologists to be, so that by the time we're in October, it will be all of us individually coming with our researches, coming with our ideas to discuss, because it's going to be a fully scientific conference. So that's the purpose of our gathering here this morning. We want to especially thank NAFDA, who is now represented here. The DG is being represented by one of the staff. We're excited for you allowing us to use your facility for this program. And secondly, we also note that even in NAFDA, we have a retinue of our members as members of staff of this organization. So coming here is like hope to have this seminar. Just to give us a little light, as I have begun to say, entomology cuts across diverse fields of behavior. All of us here are herbivores, omnivores. It means we eat both meat or flesh and plants. And how will we sustain plant life without insects? Because insects play a great role in pollination of plants. And also even the cleaning of the environment. So insects cut across several aspects of our life. So all of you, particularly staff of this agency, I know in one way or the other, you'll be interacting with insects. And so we are here to broaden our understanding in an aspect that the presenter is going to talk to us further. Even in public health, we have been having emergence of several diseases that are vectored by some insects. So insects play a great role, and so coming with this open mind, particularly that we are looking at issues of biotechnology, issues of genomic changes, and so on. So it becomes very critical for us scientists to upscale our knowledge from the basics of identifying insects and knowing insects, even going into some other areas of biodiversity. Having said all of this, I want to say thank you for all of you coming, and my wishes and prayers is that you will enjoy the seminar, because I know it's going to be interactive. Thank you so much, and God bless.
Thank you very much to the chairman of the Society of Nigeria. We'll be having the Director General, who will be represented by his CA, Mr. Patrick, also going to give his opening remarks in a bit. I think he's on his way now. In the meantime, I think we'll just maybe hold on a bit till the CA comes. So, in the absence of the PG, we'll just do a brief introduction of everybody sitting here. You can just give us your name, institution, and where you're from. Thank you.
Man Thomas by name to Anabi. My name Man Thomas. Oh, yes. Thank you.
I'm Gideon Mega, case of Dr. Konami from the University of Abuja. I'm Showman from Biotech, a member of the Ecological Society of Nigeria. Thank you. I like from NABDA. Kimotio from NABDA. Sunday from NABDA.
Correct. Thank you from NABDA.
School prefects.
So I do not University of Abuja. Paul David Eliel John. Yes, I am. Jacob Oyan Emmanuel. Let's see today.
Friday Florence. Okay. Really nice. Nina from NABDA.
Lucky Utobakala from NABDA.
Kabiru Suleiman by name of Uni Abuja.
Anita Abdeta NABDA.
Isa Mohammed Awad, University of Abuja. Obi Martins, NABDA.
Titi Lion from NABDA. Fatima Danchako from NABDA, also an ECM member. Zainab Debbie from NABDA. Awad Kasim from NABDA. Adiza Danchako from NABDA. Awad Yusuf from NABDA. Zainab Abdulrashid from NABDA. Gloria Olukono from NABDA. Achukwu Gameka Gladys from NABDA. Henrietta Chizoba from NABDA. David Uni Abuja. Dr. Teresa Olani Olushola, Veritas University.
Thank you. Let's go for it. We would like to also officially welcome the Director General, who is represented by TA, Mr. Patrick. So you can go for. Okay.
Good morning, all. Please permit me to stand on the existing protocol. The Director General and CEO of the National Biotechnology Research and Development Agency would have loved to be with you for your July engagement. So, it's with you spiritually, I'm here physically. I'd like to welcome you to the Ecological Society of Nigeria Abuja pre-conference workshop on biotechnology, genomics, and quantitative ecology. Our visitors, and we are the hosts, and staff of NABDA I wish you a fruitful and enjoyable session. Thank you all.
I'd like to say Dr. Patrick, sorry, instead of Mr. Patrick, apologies for that. I'd like to welcome Mr. David for the citation of seminar. Mr. David.
Good morning, everyone. Please permit me to start on the existing protocol. I have the privilege to present the citation of my brother and my friend, Dr. Gideon Deme. Can we put our hands together? Dr. Gideon Deme is a research associate with Case Western Reserve University, Ohio, in the United States of America, where he is conducting research using and developing various quantitative and mathematical modeling frameworks to understand how biodiversity trends and patterns are responding to the rapidly changing anthropogenic climate. Before joining Case Western Reserve University, Gideon was at the Chinese Academy of Science, where he obtained his doctoral degree in ecology, Masters of Science in Zoology from the University of Abuja, and Bachelor of Science in Biology from the University of Abuja. Gideon has published widely in science index citation journals, which have made him to be invited to be an external reviewer to many internationally impactful journals and grants applications. He is an associate editor of Ecological Solution and Evidence, one of the leading journals in ecology, and was recently appointed as the member of the Equity and Diversity Advisory Board of the British Ecological Society. Can we put our hands together?
Thank you, Victor. Yeah. Good morning, all. Sorry. Can we go back to the project? Okay. I'm excited to be here to give this pre-workshop or pre-conference seminar symposium lecture. I was invited by my mentor and boss, I always call him my boss, Professor Alan, who was my undergraduate advisor, master's advisor before I was carried away by another organization to go do my PhD. We are facing a lot of problems in this era of humans' interference into the ecosystem. And that is bringing challenges where public health is now deteriorating. We are having a lot of food insecurity. We are having a lot of climate issues. The world has moved on to use 3 approaches to solve their own problems or to managing these issues, because I don't think there's any country that has solved these problems. But a lot of the developed countries are managing these issues. And the 3 approaches that we are using now involves biotechnology,
genomics, and quantitative ecology. Now, quantitative ecology is divided into three. We have the theoretical, we have the computational, and then we have macroecology. Having looked at the fact that the ESN will be hosting one of their annual conferences, we wanted to look at these three issues as the new perspective that we can start using in studying our plants insect relationship. Can we go to the next slide, please? As a way of briefly adding to what Victor had already said, I'm a biologist by training, and my research interest involves evolutionary ecology of life history traits. I also do macroecology, macrophysiology, and biogeography. I was 3 times named the Black Man in History by the British Ecological Society. You can always, I've given a lot of lecture that you can listen to how we can tackle some of these challenges. And I'm also an associate editor of the journal that has been mentioned. Yes, ma'am. Now, here you can see that studies have shown, or we are beginning to have an understanding, that the trajectory of the Earth system in the Anthropocene is going way beyond we thought. We are beginning to have a lot of issues, increase in public health, like I said, insecurity, and we try to understand why are we facing these issues. In the last 12,000 years, people have shaped greatly the ecosystem, especially the terrestrial environment where we occupy. Next slide, please. And so, how are humans then shaping the ecosystem? We do that by modifying the equivalent diversity of species in diverse ways. I try as much as possible not to be more technical, and so I'll be explaining some terms here in case we have people that are not real course this year. So when we say the equilibrium, what we are trying to say is where we have species at the optimum, where the ecosystem can carry them and there will be sufficient services of the ecosystem. However, we are depleting the resources through habitat degradation. We are increasing the speciation and extinction rate of individual species or populations or communities. We are modifying the ways these organisms are using their resources, because when climate change is changing, they will have to be modifying their life history, their morphology, and that will also mean that they will have to adapt to the new environment. And so that has led us to begin to experience what we call the 6th massive global devaluation, which is caused by the Anthropocene or the intervention or the interaction of humans with the ecosystem. And here you can see that we have a global wave of decline in insects. So I pick out insects because this is geologically related. And we have species across different groups of insects experiencing decline. And if they are experiencing decline, some will be experiencing speciations, trying to adapt to the new environment, thereby bringing in new diseases that we are experiencing, or new bacteria that are emerging, or new challenges that we will have to deal with. Next slide, please. Now, if we are having changes in the ecosystem or the population of the species, it means ecosystem services is declining and our well-being is at stake. If you look at seed dispersers, if you don't have birds
or insects that will feed on the on plants to pick out the proteins to disperse the seed. Then we will have decline in seed dispersal. We will have decline in decomposition. Our water quality will be deteriorating. We will have decline in pollination. Our carbon cycling will be in a mess, which will cause the ozone layer to be depleted. Now, one thing that you don't know is that species interact with each other. So aside the abiotic component interfacing with individual species, we have a biotic component. And that is what we now call the species network. And to disentangle some of these things to understand how are these species moving and interacting with each other, you need a lot of mathematics. And that's why when I was an undergraduate student, we were always told that if you are in biology, forget about maths. But I learned it the hard way. Because if you have to solve global issues, whether in biotechnology, you need bioinformatics. And bioinformatics, you need mathematics. Now, you have species sorting out themselves as they are interacting. When a species goes into an environment, it has to deal with the environmental challenges. And it either has to establish or exit. So that is what we call the entry and exit rule in ecology, where a species comes into an environment and it adapts or it undergoes plastic development. So plasticity is where it will just be a rapid response that will be affecting only the phenotypes and not the genotypes. That means they cannot transmit those traits that they have gotten in the environment to their offspring. So if it's a plastic response, that means the offspring will have to have issues. And as species are continuing to sort themselves, the fit ones will remain. Those that cannot fit will exit. Either you exit by force, or either you exit voluntarily or you are exited by force. That means death will exit you. So we try to understand if we have a lot of these issues, how then can we bring some approaches that can help us understand? Now, this is what we call the holistic approach to the new issues that we have, where I was talking about biotechnology, genomics, and quantitative ecology. Now, that is a schematic diagram where you can see almost everything represented. We have our microorganisms there, we have our herbivores, we have our pathogens and plant diversity, all interacting. But if you look at the top, environmental pressure, such as climate change, pests, that are being introduced by humans, are having impact. Then you have to have a multidisciplinary approach to solve this issue. And when you talk about quantitative ecology, like I said, you have the community ecology, you have the macroecology, you have phylogenetics, which is molecular ecology, or evolutionary ecology, you have the evolutionary genomics, and then you have the biotechnology. So if we in Nigeria can begin to marry these approaches, we will begin to have to deal with, because one thing that we need to understand is that biodiversity interacts with food and climate. That is what we call the biodiversity food climate nexus. You cannot disentangle this. And so we need to develop ways to tackle these challenges. And so I will be giving you some of the studies that I collaborated with some people to do using some of these things. But I don't think I've got any biotechnology sequencing because I probably love working on the computer. But I'm collaborating with a lot of biotechnologies. Just like this. Now, we have biotechnology approaches where we use the biological enemies of the insects to tackle them. Somebody here was talking about the Bacillus thuringiensis bacteria that is being used. I can't remember, but yeah, these are some of the toxins that are being introduced to control pests without having to use pesticides. And so when you introduce some of these biological weapons, what they do is they have a way of attacking the neural system and the biochemical system of the insect and naturally exit them from the environment instead of using pesticides to spray and introducing chemicals that will have harmful effect. Another way of that people have introduced is the genomic editing, where you can actually delete or add a genome to modify an organism. And so you have a lot of these advanced, I believe I cannot mention the people that are here on genomic editing. You have more insight into how this genomic, but for the purpose of people that are here that might not have, you target a particular gene where you feel if that is the gene of the problem, you target it and delete it. And then there is a way that you now have a new organism without that particular gene. If it is a gene that, okay, you understand that this particular gene is what is making the mosquito to be able to carry the oocysts of the parasite, you target the gene to remove it, and the mosquito will exist but will be barren to carry the parasites. So these are some of the ways. Another way is where people are beginning to do the RNA interference, where the RNA is suppressed. The RNA of a specific gene is suppressed. Assuming it is an insect pest that you want to get rid of, you suppress the RNA that enable it to survive. If you want to increase their population, you cannot suppress the RNA that the environment might be targeting to modify. So it's just a way of maxing a particular genome area that people have developed. And some of the things that they use is what we call silencing mechanism of the double-stranded RNA at the cellular level of the organism. Next one. And then we have the marker-assisted selection in biotechnology, where you develop a lot of primers. And when you develop, just 2 days ago, I was sent an email to join the global scientists that we develop. One thing that we have problem in the tropics is accessibility to some of these things. So the scientists want to develop what we call global primers that you can now use and modify into your own kind of organism or into your own kind of primers that you want, but they will be globally available. So once you have the primers like the single nucleotide polymorphin, that's the SNPs, you select for the phenotype that you want to improve or you want to remove. So you control the phenotype from the genomes of the particular species. Aside the you develop a phylogenetic tree to understand how our species relating in time past and what will impact their relationship in the future. So things like, so if you want to understand, okay, how can I make a plant to relate with an animal? Because it's not every plant that insects will interact with. So you have to study what are the plants that have evolved at the same time with species of particular insects that you want to work on. And another thing that you have to look into are the phenotypes of the plant. For example, the emergence period of a plant and the emergence period of an insect will have to synchronize. If they are not synchronizing, what will happen is that when the plant will be flowering, the insect will be at the larva stage. And so there is no relationship. And so when you use this kind of approach to try to understand how are plants relating with species of insects that you are targeting, then you'll be able to have an understanding, and you will now also understand, okay, if climate change is impacting these insects as they are migrating, are they able to track back these plants because plants are mostly immovable. They move very, very slowly, but insects can migrate faster. And so you can have plants that the plants now, the insects have gone. But these kind of studies will be able to enable you. So what we call that is in biogeography range shift analysis, where you are able to understand the tracking of where the species are going, and if they are able to trace back their origin to be able to interface with these plant species. Exactly. And so evolutionary genomics, like the genome-wide association studies, are good for these kind of studies. It allows us to explore I was just talking about the evolutionary transitions. The plants beginning to develop some kind of chemicals that the insects are not liking at this point. How then can we modify? How can we make them to interact? Because if they don't interact, we'll be having issues of food security. Because pollination will be at the lowest form. That's like it. And so some of the interactions like I was just talking about is where you simulate for the plant chemicals and the genomes of the insects to try to understand the relationship and how the relationship can be maintained in going forward. Because now, when you have this kind of, when you build a phylogenetic tree, there's something we call climate modeling, where you can now model climate, future climate, on the phylogenetic tree to give you an understanding, okay, in 100 years to come, where we'll be having a 1 degree increase in temperature, what will be their relationship? If there's a disentanglement, what will be the cause? Is it that there will be more release of some chemicals that will make the insects to be repelled? Then you try from now to control how these chemicals can be by reducing their genes or reducing the amount of the genes at the cellular level that will make them come out overexpress some certain chemicals that are repel the insects. Next slide, please. Maybe a little time for me to have my cough. Maybe you can just give me, I can share the slides after the lecture. So I can just use your computer.
Because we're online, so I don't know what we can do. Are you just stay by this side?
Yeah, I can just stay here.
So you can control yourself.
Yeah.
So we are online, so I want online people to participate.
I hope everybody can hear me. Yeah. So, apologies you can't see the slides, but I'm happy to share my slides after this for those that want. So, the next thing that I was talking about is where we use high power sequencing to understand, because it's not every kind of study. Now, when you want to develop a phylogenetic tree, it depends on what kind of study you want to do. There are studies that if you use the 16S RNA, if you use the 16S RNA, they won't give you the information that you need. You might need to do DNA barcoding and other stuff. This thing is killing my side.
I will control the slide for you. Maybe you can move forward.
Do they have to see me?
Yes.
No, I think the slide is the most important.
Okay, it's nice. So let me just off the video.
Yeah, because right?
Okay. Yes. So
but they are online. But I mean, the battery is running down.
It will stay for some few seconds. Go ahead then. I will switch off in second one.
Okay. All right. Let's continue. And so, what I was trying to say is that it depends on the kind of sequence or the kind of questions, and that is the biggest issues we have in our Nigerian system of studies. We just go into studies with no questions in mind. You just assume, oh, I want to study the prevalence of malaria in relation to mosquito abundance. What is the question? Because if you have a question, it helps you to collect the kind of data that will address those questions. It's not every data that answers every question. For example, especially if you have to use phylogenetic questioning type of system, or you want to use an evolutionary aspect. You can do that in maybe superficial baseline studies, but when you want to go into deeper issues, when you want to solve problems, you have to have a question, and then that question helps you to design your experiment properly, and then you'll be able to do your analysis with the data that you have. Just put it on the slide. Okay. Okay. So now we are, as I was saying earlier on, that species are moving. They are not staying in one place. And it is because climate change is impacting. And as I it might be impacting body mass indirectly through the impact of host plant. That means if you are feeding or if you need more host plants and you have more continuism, you'll be able to have a smaller body size or smaller body mass to have a wider range size. And so due to these assumptions, now we plotted or we figured our structural equational modeling that I was talking about, and then we brought these figures that you are seeing. I'm trying to be less technical so we can all follow. And you can see, interestingly, that body size was related to range size. That means, and here also, range size was, so some of the traits that were not impacting directly were impacting indirectly. So if we had just done a straight-jacket analysis, we would have been missing out some key information. And so that's the importance of the multifaceted analysis to be able to determine, to go to the details of your findings. Next slide, please. The next question then is, okay, let's even forget about genomics. Let's go pure into quantitative. Let's assume that, okay, butterflies are just clustering as friends. We all have friends that we were never related to. We migrated from Sokoto, KB, Lagos. We all are in Abuja with 4 friends. We were never, so it's not everything about genomics, biotechnology. It has something to do with mathematics and associations. So we use some survey data of butterflies, fruit-feeding butterflies, and then in this context, it was community ecology approach where we try to understand if the differences in the species that we see across habitats will be impacted by environmental conditions, or if habitat with different heterogeneity will be able to influence the abundance or the richness of butterfly species, and if the local scale butterfly diversity is impacted by the urban landscape micro geographic differences. And that is, so what we are trying to say is that you have Biotechnology Institute or Research Development Agency is in the city of Abuja, but the landscape here is different from the University of Abuja landscape. And the landscape in the University of Abuja is different from the landscape at the central area of Abuja. So can these support different types of relationships between butterflies and their host feeding plants? Yes, we found a great diversity. And you can see the non-metric that statistics showing a strong impact that, yes, you are in biotechnology, I am in the University of Abuja, we can support different systems of plants and their pollinators and butterflies. Next slide, please. Now, you will see the clear differences, right? Where we use the NMDS, that's the non-multidimensional statistical analysis to show which habitat supports more and which habitat supports less of diversity. Next slide, please. And then you can see that the savanna, which have different plant structure or vegetative structure, support more of the butterfly species. So what that is teaching us is the fact that even if we want to have a plant-insect relationship, we have to have the knowledge of the vegetative structure. If you just go to just plant any kind of trees or grasses, thinking that, oh, I want to invite more insects or to have more population, you are just living in a dreamland, because these insects are not as emotional as you are. They won't say, oh, it's because Mrs. A has worked so hard, let me go visit their garden. No, they are selfish about what will be my benefit if I go there. Just like that. And so that is what I've just summarized. The next slide. And so aside that, we also look at the mathematical angle, right? That okay, this study that I just showed you, we used a smaller sample size, but we have the biodiversity records are running into billions of records, like the GBIF, right? The Global Biodiversity Information Facility that hosts a lot of records of biodiversity is currently at 3.6 billion. Although in Africa we only have about 4.6 million in contribution, and Nigeria, we have just 800,000. We need to do more. So we try to understand that across the spatial complexity and the temporal complexity. So let me try to break down a little. You can see that we have two different landscapes here. We have the larger landscape and we have the smaller landscape. What is going on here is different from what is going on here, right? And in mathematical ecology, we have two types of theories. We have what we call the neural theory or the neutral theory, where we assume that, in habitats, nothing is going on. Everybody is given fair opportunity. I don't think that's possible. But then we don't have the other one that we call the deterministic and the stochastic theory, where we talk about natural selection, things we can control and things we cannot control. So the deterministic are the environmental conditions, while the stochasticity are the non-determined things that we cannot control, like flooding, like death rate, like birth rate, although we can control birth rate, but technically we cannot in a way, right? So these are stochastic conditions that we assume can impact a relationship. So we assume that, so as you can see this slide is back again, where we are talking about the associations of species, when you come into a new environment, whether you exceed or you still retain. Next slide. And then, so in this place, can you click? No. Oh, so this was supposed to, in my computer, there are some mathematical theories that are behind it. But it's like your slide is not supported to show the equations. Let's go back, let me just explain briefly. Yeah. So here, we have a mathematical theory behind what we call habitat suitability, that the suitability of a habitat will be what will be explaining the relationship between different organisms targeting the same plants. And in this instance, we assume the birds and the insects. In the context of Nigeria, I would say the songbirds and butterflies. But this study we did in the US, so we use the hummingbirds and the hawkmoths, where they relate and share the same habitat. So what we assume is that niche partition, where yes, we are neighbors, but you are strictly on your own. I mean, we have instances where you don't even talk to your neighbor. So this is where we talk about niche partition, where we live in the same habitat, but we assume that this person does things independently. And this is more so why we use the hummingbirds and the hawkmoths was one is a diagonal, one is nocturnal. So we assume, okay, you come and feed in the morning to afternoon, I only come and feed in the night. So we partition our day. Or we can overlap where we assume at 6 p.m. where it's still within the same right, we are still there, and then I come, we overlap. Next day. And then the last one, we talk about the carrying capacity of the habitat. If you have a smaller habitat, then we will have a less interaction, or if you have a bigger, or more interaction, if you have bigger, people who have their species to interact with the plants. And so I've already talked about where we use the hummingbirds and the hawkmoths to, let's say, we try to measure the extent at which both species will interact with the plants at the the same time, that is core or to understand the pattern of the trend. So if they are interacting, are some declining in their populations or they are maintaining a stability in their population, and then to assess the mechanisms that influences the distribution. The next slide, please. We found evidence for a strong relationship. I should say, in this case, we used the generalized additive modeling where we assume it's a non-linear relationship, right? It's not all about I directly, I can be indirectly related. So that these are the kind of models I give you, and that's why you see the curves are back a little bit. That's why you see the curves are not straight in some instances, telling you exactly how the relation. So what we found is interesting that in the tropics where we have found, where we got plants, you have a lot of them associating together, right? But once you move to the temperate region along the latitude, they are declining in their relationship, but in their abundance and richness, but their relationship is still strong. This. However, when you go higher elevation, common birds tend to partition their niches. So it means that our two assumptions were proven right? Because once you begin to go to higher elevation where you have less resources and extreme conditions, then common birds take advantage because some things we don't know. I don't want to speculate because we are still trying to investigate. Next slide, please. And then we look at their relationship with the deterministic, which are the environmental conditions. And we found that colder temperature and drier conditions will always reduce the availability of resources like plants, as we talked about. And that will negatively decline the relationship between plants and animals. That means climate change is having an impact in the plant-animal relationship. And then we talk about the diversity across the elevation. Next slide. Next slide. So, if assuming all of these things are very, very time consuming, right? Because you have to do some mathematics, you have to go. We have developed AI now that is being used in biotechnology, in genomics, and in quantitative ecology. And I'll just go. AI have been developed to be able to identify proteins within a space instead of it. So you just have to train that AI with a little bit of a protein, and it does the remaining identifications for you. That is the level at which the world has moved at. In terms of genomics, it's the same thing. You just have to train the AI on some of the genes that you want it to identify, and then it does the remaining identification. Because now we are beginning to have genes of millions of reads. You have cellular language that RNAs, millions of reads. The studies that I just showed you now are dealt with hummingbirds and hummingbirds. We use 16 million records. Imagine you were to identify public health, food security, biodiversity. And in this sense, when I talk about biodiversity, I talk about plant animals or interaction between insects and animals. So if we don't apply these things, and recent studies have shown that the increase in and the outbreaks of all the diseases we are seeing have an indirect relationship with urbanization, that is human footprint. The lack of using these modern tools, right, is draining our economy and even our public health. So as scientists, we need to care. But how then do we care if we don't have money? For those that are in the universities, I know how hard things can be about funding. Next slide, please. I decided to put up some funding opportunities that ESM and anybody in biotechnology can keep into. For those of you that don't have a PhD yet, there is the Newton Fund that is available in the UK for minorities like you. You can apply and then you have unlimited amount of money to do a lot of this research. The Wellcome Trust, if you have a PhD, you have the ability to get £450,000 to drive your ideas. Just make sure that you have good questions in mind, because as a reviewer of the grant, I'm always interested in that question. And do you have the methods to answer those questions? We also have the Darwin funds, which can range from £600,000 to £5 million. Let me tell you, let me run through this and then I'll close with this. Then we also have the Career Development Fellowship of the Royal Society. This is only for PhD holders that you go to the UK and then collaborate with one of the universities. But your research will be basically from where you are coming from. Okay, I forget that some people are still looking at me. So we also have the British Ecological Society has various types of funding. We have experts on African-focused research that is hosted by the University of Helsinki in Finland, which is also only for PhD holders, and then we have the National Research Funds of the Tech. Let me quickly speak on the British Ecological Society. As a society, so I recently joined the highest decision body of the British Ecological Society of Diversity and Equity. And one thing we want is local societies in the tropics to collaborate with the British Ecological Society so you can be funded to tell us your own story in your own comfortable language. So if the Ecological Society can write a proposal, and so we will be meeting in October. We are discussing about some of these things, like what are some of the vibrant societies? Because we don't want to deal with individuals. We want to deal with a society that can reach out to a lot of people. This is where we can leverage on. The British Ecological Society makes a lot of money from the publications. I can tell you from the fact because I'm an associate editor. So it depends on what you want to do. How do you want to enrich insect ecology for the broader group of? You can even make it not only about Nigeria, but about West Africa. Since you have a long history, the experts in Africa or Africa Focus is where you go to this university and then you are getting a lot of money to do a lot of research for at least a year, right? So these are some of the opportunities. But the bad story I always say, or I always love to tell you the bad story, that you have to fail and fail and fail. They don't think because it just happened once, you will be funded. Nah. I was telling Victor that behind one success story, there are 10 things. People will not tell you. So, what I'm not trying to say is that if you keep applying and you keep rejecting, keep knocking on the door, there are a lot of funders. All you just need is one door to open. And then you can do your research and be a global scientist. And once you get just one of these funding, then many will open. They just want to know, can I trust this person's idea? Yes. Once they see on your CV that you've once won a welcome trust fund of 450,000 pounds, and you did the research, nobody will be scared of giving 5 million pounds because you've proven your worth. On this note, I end here, and I want to acknowledge my collaborators who are in the US, the organizers of this event, and the Nigerian Biotechnology Development Agency for providing this space. Thank you. I'm happy to take questions.
Thank you very much, Dr. Gideon. We'll be moving to the interactive section. Answer questions. So let's move to the interactive section. I'd like to entertain any questions from the audience or from our viewers online as well. Any questions, please? Observations?
Thank you, Dr. Gideon, for your presentation. The question is, you talked about AI.
Okay. Yeah, I'm listening.
Okay. You talked about
questions? Annabelle?
Okay, so my question is that he talked about the AI also. So I want to ask, how long do you need to train the AI before it can start working in terms of identification?
Thank you very much. Let me just take a step from the AI question. For me, it's which particular AI applications can be used for tracing insects or birds. The other thing I wanted to draw our attention to is he made a statement during his presentation. Thank you very much, Dr. Gideon, that we do not ask questions when we're carrying out our research. I don't know if it's an issue of the way you look at it. Maybe the way we do research here is different. I know that when I was doing my research, we do what we call hypothesis, research hypothesis. And your hypothesis comes from the questions that you're hoping to answer. So maybe it may be the same thing we're doing, or in a different way. So I think the bone of contention will be to encourage us to either do an hypothesis, because that is when we can actually tailor our data to answering these questions. Thank you.
Another question?
Another question?
Okay. My question is that you mentioned of environmental adaptation, and according to Charles Darwin, when animal went on undergo a environmental adaptation, they changes their shape. For example, a giraffe was a short neck before, according to Charles Darwin. It's because of that environmental adaptation, and it's later came with a long neck. So my question is that does the same thing happen to plants and animals also? Thank you, sir. Sorry, the mic is not so thank you, sir.
Thank you, Dr. Gideon, for your presentation. I can go on and on. I honestly understood a lot from what you presented. I understood a lot from the biodiversity, the ability of insects to adapt to the environment, and also the dependence of the environment on how they adapt. And if they are not able to establish, they will exit, and that's what you said. I really appreciate this presentation. And in terms of a question, I have two questions, right? The first question I have is concerning the use of AI, right? I don't want to create any confusion. I know that there's something that is called virtual reality, right? Virtual reality, where you could be in another place and then we'll be communicating, and using virtual reality will be able to help me achieve some things, right? I want to know if there's something that we can be able to adopt in these technologies that you just clearly spelled out, right? And then the second question is not more like a question. I want to ask if I can contact you personally, because I'm also in a group that is looking forward to making a product regarding insects. And based on the information on your slides, I can really tell that you are really grounded in this particular field. And I really want to, if not collaborate, just want to inquire one or two personal questions. Thank you.
Thank you very much.
Can I answer the next question? So we have the next one. So there are a lot.
So let me answer this questions because there are a lot. It meant that we were all together, and I love them. Very brilliant questions. Dr. Teresa is bringing a very interesting debate. Okay, so the first question is where Victor was doing a reality check, as if we are really ready in this part of the world. Well, I'm an academic. Sorry to say, when you say ready in one context, but I don't want us to go into a question and answer session. I'll just tell you that, yes, we are, right? And if you say we, I hope you are not looking at the collective government, no. I'm not here to tell the government to come in because that's the whole kettle of fish. Tell the government to really give you 5,000 for research, you have to debate for 5 hours. I'm not here to do it. But if you get this, so when are we ready? Yeah, you are ready because you can learn it. You learn that you are applying. You have your computer, you power your phone, you do your research yourself. Dr. Olaruwaju has continued to do a research even without the government money to get to. I mean, it's only Nigeria that you have to do your personal research from your money to get to a position. Right? So people are beginning their personal help. So yes, we are ready. We are ready. And that is the mindset that we need to reorient ourselves, right? Because the world is moving. The world doesn't really care if you say, I'm not ready. People will keep moving. And you have the issues here to settle. right? So yes, we are ready individually. Or let me say people that want to advance in their career are ready. Right? Because it's all about learning the procedures and the process. And you can do it. Somebody is using ChatGPT here. Right? And then people are using ChatGPT, that's here, to write their speeches, their articles. We are ready. That's here. We are ready. I don't want to fall into this category of we are not ready, because that will dampen the mindset of the law. Right? A lot of people here know my story. Where I came from, Dr. Ecology knows me from far back 2008. So she can tell me the struggle, she can tell the color of the one shirt I was wearing. Victor, you know me as a person. So I didn't emerge from the US to come here. I was born and brought up here. That's why I used to change my accent. Because I want to be in Nigeria and I'll be in Nigeria. Right? So we are ready. It's about the mentality. We need to switch your mentality to say, yes, I'm ready. And keep into the global perspective. How long does it take to train here? Now, how many of you have used machine learning before? Machine learning is like AI, right? So machine learning in modeling, right? I've done some machine learning for simulation. It's just a small, it's not about time, right? AI is not a human that has a brain to think, right? So in machine learning, what we do is when we are modeling, you pick out maybe 20% of your data, of your what we call crowd data, right? Then you run a model. Now what the computer does is to store the information and then use the remaining, the information from that 20 and like, okay, Mrs. Hadiza looks like this. So if I see anybody with this facial look, this is Mrs. Hadiza. So it now does the remaining 80. It's not all about time. It's a split second. It has no brain. It is algorithm that recognizes things. So you just develop your own algorithm and you use a little bit of data and train and inputs. And then Dr. Teresa asked about which there are different types of AI, like virtual reality is what is used for population. But I try to remember the one that is used for biotechnology, right? There are differences, but I apologize. I don't know the thing because there are a lot out there, right? Even for synthesis, do you know that we use AI now, aside ChatGPT, we use AI now, which we call language modification AI, to be able to translate a literature in Chinese back to English for you. So that, assuming you want to do what we call meta-analysis, where you want to do a global synthesis of literature, you can download literature from China, which is written in Chinese. The language AI will translate it back to English, and then you get the information. Right? So the virtue, you train the virtue with the kind of insects that. So it is one part of it. You just, how many of you have, I was introducing this master student to SIG. This SIG by iNaturalist, right? Once you just slap the butterfly or any species, it identifies for you and even give you a historical background where it came from, if it is an introduced species in Nigeria or an endemic species. It's all about algorithms. I have it on my app, so, and you know something, let me tell you a little bit of a story. That we Nigerians don't know how to sell ourselves. Now, you might be able to know how to use a seed, but in your CV, you don't have have the proficiency using AI to identify this. That's where the white man is fascinated. If you can use it, that is you having the ability to use AI to identify species. You just snap and it identifies you. In your city, while you have the knowledge of AI to identify your all come and work. So that is the kind of AI that you use for insect too. It's an app. You can go to your Google store. Just seek by naturalists. You will see. I gave this, right? I downloaded it for you. SEEK. Okay. You just seek by natural. If you use an iPhone, go to Apple Store. You seek by naturalists. You just snap whatever it identifies for you and tell you the historical perspective. Now, Dr. Delika said we use hypothesis. Hypothesis are also of bachelor's and master's. So I made a categorical statement. We have our objectives. Yes, we can assume that's questions, but those are questions. So research are able to right? We have the objective. For one objective, you have 3 questions. In those 3 questions, then you now have your hypothesis addressing each question. So you have an objective, then you have questions, then you now have individual questions with their hypothesis. So we have this missing link between objective and hypothesis. It was a deliberate statement. I know it will instigate a debate, but I'm always ready for a debate. Yes, but we need to have questions addressing those, because one thing we think is that one objective can be addressed by one set of data. Now, one question can be addressed in 3 different ways, and I will give you an instance. So, if you give an instance that, let me go here. If you give, let me see, the objective of this study, one, is to determine the socio-economic factors that impact mosquito prevalence. Socio-economic is broad. Then you can now ask the question, can income as a socio-economic factor impact mosquito abundance? Now you are pulling out income out of socio-economic. Then you go to educational status. What level of educational status can reduce the abundance of mosquitoes in a household? That means if you have a PhD, do we expect you to have the knowledge to be able to manage your environment to reduce the problem of mosquitoes? That is a social economy. So when you bring questions to address an objective, then you now bring hypotheses, both null and alternative, to now address each question. Where you now assume that, okay, income will not be able. No. If you have higher income, you have a beautiful house, you make a good garden, mosquitoes will not be there. So we do a job from objective to hypothesis. But we can have that debate together in our personal time, not in a gathering like this. So I hope I've addressed that. If you are not satisfied, I have my number, we can always check. She was my senior in the case. Right, so there was a question whether plants do follow the rule of natural fitness. Let me just dispel that Lamarckism that you talked about. It's not true. There is no scientific evidence that shows that a giraffe will straighten its leg and that's why we have giraffe. No, it's not true. It was just Lamarck being crazy. We love scientists that are crazy. It has never been proven. But yes, plants do follow the rules of natural fitness, right? Natural selection. And I was just talking about the phenotypes of the plants, right? Even when the plants start to flower, it can change due to climate change. The traits of the plants can change due to climate change. It doesn't have, even the groundwater can impact the movement of the plants. That's why you see some plants bending towards the north or towards the south or going straight forward. All these, yes, they follow the rules. There's no species that natural selection doesn't impact on. But we have species that can be able to resist natural selection. And then the ability to recognize the impact of natural selection is negligible. And that's why if you change location, you see your skin color changing a little bit. You change location, you see yourself giving birth to children that are growing taller than you. And then you trace back, none of your grandparents were that tall. That's natural selection. Yes, you can contact me this. And the visual reality is one of the eyes that we use. Thank you. I hope I've answered all the questions. I don't see how this is.
Thank you so much, Doctor, for the seminar. My question is that, let's say if one is working on, let's say, transgenic insects, for example, such studies, will it affect the ecosystem?
Transgenic?
Yeah, that's one question. And I don't know if you have room for collaboration.
I don't have room for collaboration.
Okay.
That's me, that's me.
No, I said if you have. No,
I don't think
you don't have.
That was making a categorical statement.
Abraham, a question.
I've collaborated a lot too.
Thank you so much, sir, for this very. I'd like to ask if Google Lens had the same capacity with the seek by nature, because when I was trying to identify some of the insects last year, I used Google Lens and it was very, very effective. I think except for some insects that we get size. So then secondly, I would also want to inquire if the Shannon diversity index during those visits are very effective in our assessments. And then lastly, we did the work on the biodiversity assessment for transgenic people. So many stakeholders who are not really sensitized about the crop were like saying the transgenic copy would have an effect on biodiversity. So I would like to find out if something similar has been done before.
About what? This
like if certain transgenics will have effect on non-targeted species in an ecosystem.
Certain
transgenes, that is genes
yes.
started from, will have certain effect on non-targeted species in an ecosystem. Thank you.
Any other question? No other questions?
Thank you. Anywhere I go, I always say Nigerians are extremely brilliant. And if you are not prepared, you will pay for the price because you will get challenging questions. To start with, can transgenic insects impact the ecosystem? Negative or either way, yes. And that is why when you are doing your transgenic of whatever species, and I come from the world where we use a lot of genetically modified food species, but you have to put into account one, that modification they are making for insects that you ask, right? Are they now going to become susceptible to pathogeneity, susceptible pathogeneity, or are they not? If they are, can they not transmit? Because there are some transgenic insects that will not become vectorial. If they don't become vectorial, then are they impacting their ability to fly for pollination? So if your transgenic insects reduces their wing size, thereby reducing their flight time, flight period, then you are impacting the ecosystem negatively because you have less population. And that's why you always see scientists that want to make transgenic insects to improve population, we always make those with bigger wings to be able to travel far. However, their weight, that wing size does not add to their weight to destroy the flower when they are pitching on. Right? So, and that's why a scientist that is careless is worse than a battalion of army. Because one finding can rule the whole country. What brought about terrorism? It was one man that discovered AK-47. And today nobody can see with their eyes closed. Right? So they have an impact. If only we don't meticulously do the things that are supposed to. And that's why we don't just sit in the lab and just transform an organism, right? You have to ask, why do I need to transform them? What would be the impact? What if it exceeds the impact? How can I control it? Right? Because, yeah, you might be able to do it thinking, okay, there might not be, but you need to put a control mechanism that, okay, in case I transform Gideon to be able to walk 50 kilometers, but somehow Gideon exceeds to 51 kilometers, he's going into somebody's farm. How can I, what control mechanism? So scientists out there already have control mechanism for in case that's why you see they quickly recall back and remodify. So it can have positive and negative. Go, I cannot say, I'm not that kind of person that wants to compare options, but I think I've used this. It is more detailed. It gives you, Google Earth, but there are pros and cons, right? Google Earth will give you even the coordinates, right? Seek will not give you the coordinates. But you can extract the location and they'll find the coordinate on the map. But Seek will give you the entire families, whether this was introduced, this is endemic, if it is introduced, has it become invasive? Or is it still within the population that it was expected? These details, once you start, you will see a whole lot to it. Right? And then you'll be able to now extract the location and then use Google. I've always preferred Seek because it is very, very detailed on the taxonomy, the life history, and even the relations, the evolution of whatever. It gives you those kind of detailed information that in case you want to, because if you have your findings, you need to discuss new things. So these kind of information are those things you put in your discussion, and that's how you will cite them. And then, my brother Takanashi. There's a debate in community ecology where we think shallow index is just a wash. That's not shallow index I'm sure in my studies. Shallow index is only good if you want to just understand whether it's evenness or richness. Right? But if you want to know how much of the species are changing over time, which is trend, then you either go into beta or gamma analysis, right? Because shallow index, we believe, is highly biased. Like if you have 50 species that have 50 occurrences versus one that has 3, shallow index will favor the one with 50. You definitely see dissimilarities, but it might not be. It might just be abundance, right? So you go into beta diversity. We have what we also call the Jacob Index. Yes. Yeah. So the Jacob Index is a mathematical approach with statistics in it. How many of you remember your set theory in your mathematics? Yeah, it tells you how much of the union species are uniting or they are not. So these are some of the community indexes that we use. A lot of the viewers, once they see shallow index, they are like, nah, don't do another stuff. Go do something. Lastly, he asked, I'm finishing the last question. Effect or not effect? There could be. It might not be direct, right? So you might have, there are studies that have shown that transgenic organisms have impact non-targets. But you might also, it might not be direct, it might be that when the organism now becomes transgenic, it might now become a facultative parasitic organism. That means it can feed on another organism. It can be a facultative parasite, but can be free. So our transgenic, that's why we need to be very, very careful as we do our transgenic lab work. I will always say, put control measures in place in case it doesn't meet our expectation. We shouldn't waste another 5 years trying to undo the harm because science is risky. You cannot, you don't have the monopoly of knowledge. You might think you've done the right thing, but things like that but have the ability to control. Then I think I've done with the person.
Thank you very much, Dr. Gideon. At this point, we'll come to an end of this seminar. We would like to call on Dr. Hajara Oyusu to come for the closing remarks.
Good afternoon, everyone. I think our presenter, Dr. Gideon, deserves a very loud applause. Honestly, when you hear the word entomology, some people think that it's just for those that have to study insects or maybe chemists and biologists. I don't belong to that. All of us belong to it. And what they have done here today, this seminar, was so loaded that, and towards the end of it, I was so happy when he said, no, when we have a blackout, like outrage, he said he's going to share the document, which made it interesting because some of us might be listening, it's slow learner, and there are people that are fast learners. So at the end of the day, when we look at the slide, some of us might have a question that she also gave us the privilege that is very free to be reached out to. So I find it very interesting, and I'm so happy for what you've done for us today. And I believe the scientists and researchers in NBRDA, we talk about genomics, quantitative ecology, and it's something that our biotechnology research is into in everything we are doing here. So thank you so much for the seminar presentation. Another important thing is that the Ecological Society of Nigeria, which I'm a member, I know some of you are also members here, we need members to join because our branch here, which is Abuja branch, is hosting this year's International Conference on Entomology in Abuja by October. If you have not registered to become a member, try to register. I will tell you how important this society is. It cut across, and the benefit is enormous. So you are welcome on board to join. As I wish you, those of you that come from far to this place for this seminar, safe back to your destination. Today is Friday, we say we thank God it's Friday. So happy weekend to everyone.
Thank you very much. And conversation continues. Dr. Gideon is very much around. If we have any questions, you can always address any of them. We will also be having certificate collections after the conference. And group pictures.
I want my apology. We will not get our certificates today because it takes some time to write the names and to get them. But I will use the list that has been compiled. After that is done, I will give it to the DG, and then I think I have the numbers of all the participants. Maybe I will send an SMS to a few that ready. But by Monday, my colleagues. So we would assemble downstairs for the group pictures. Thank you.
Let's have closing prayers.
Okay, closing prayers.
Please, really quick.